Welcome to Podcasts on Demand, a continuing medical education activity. This activity includes the most recent and current clinical data presented by leading experts. If you are seeking continuing education credit, please review the disclosures and the requirements for a successful completion of the activity prior to listening to the podcast. A link is found in the podcast description that can direct you to this information.
Thank you for joining us for Conversations in Advanced Non-Small Cell Lung Cancer, Rationale and Utility of TROP2 Antibody-Drug Conjugates, based upon updates from WCLC and ESMO. During this segment, we will be discussing the impact of ADCs targeting TROP2 and other novel targets. I'm Dr. Alex Spira, co-director of the Virginia Cancer Specialist Research Institute. I'm joined today by my colleagues, Dr. Ben Levy and Dr. Aaron Lisberg. Great having you. So there's been a lot going on, and it's been many years of ADCs now, and they're the hottest thing in non-small cell lung cancer. And so it's almost hard to keep track. We're going to start today, gentlemen, by talking a little bit about the TROP2 ADCs, which are probably the farthest along. And let's start with some of the studies just reported. Tropion Lung 01. Aaron, you were an author on this. Why don't you take us through Tropion Lung 01, and then you can even segue into Tropion Lung 05. Tell us what the studies were looking at, what they showed, and kind of where we go from here.
Of course. Thanks, Alex. And you and I worked very closely together with data for many years leading to these studies. So the Tropion Lung 01 study was a large randomized phase 3 study evaluating Dato-DXd, which is a TROP2-directed antibody-drug conjugate, against our standard second-line chemotherapy docetaxel in patients with advanced metastatic non-small cell lung cancer who had previously received therapy and were eligible for second-line chemotherapy. Patients were enrolled whether they had or did not have an actionable genomic alteration. The non-squamous and squamous histology were both enrolled, and there was no minimum TROP2 expression level that was required for treatment. They were randomized 1-to-1 at that point to receive Dato-DXd every 3 weeks versus docetaxel. And the trial had two dual primary endpoints: progression-free survival by blinded independent central review and overall survival. So there was a lot of data that was presented at ESMO. Essentially, what we saw was that in terms of patient disposition, 3 times higher number of patients on the Dato-DXd arm were on therapy at the time of data cutoff compared to docetaxel. We also saw that more than half of patients treated with docetaxel were on therapy for 3 months or less. This was in contrast to 20% of patients on the Dato-DXd arm who were on therapy for at least 9 months, compared to less than 10% in the docetaxel arm. Reason for treatment discontinuation and the median study follow-up are similar on both arms. So when we looked at the first of the 2 dual primary endpoints, progression-free survival and the intention to treat population, what we saw was a statistically significant improvement in progression-free survival in favor of Dato-DXd over docetaxel, a hazard ratio of 0.75, more than doubling of the objective response rate, a longer duration of response. The median PFS in the Dato-DXd arm was 4.4 months compared to 3.7 in the docetaxel arm, though. And we then looked a little further in terms of which patient populations were deriving the greatest benefit, and we looked at a forest plot in the ESMO presentation. We essentially saw that across the vast majority of the characteristics, there appeared to be a PFS benefit in favor of Dato-DXd over docetaxel. However, when we looked at histology, there was a clear divergence in terms of this PFS benefit, where patients with non-squamous histology were clearly benefiting, and those patients with squamous did not, with a hazard ratio greater than 1. We also saw in that forest plot that the patient population with the greatest benefit from Dato-DXd with respect to PFS was the patients with actionable genomic alterations, and all but 3 of those patients had non-squamous histology. So we looked at this a little closer in some Kaplan-Meier curves on the ESMO presentation, again seeing that in the non-squamous population, there was a clear separation of the curves for PFS in favor of Dato-DXd, almost tripling in the objective response rate, a much longer duration of response, and median PFS benefit of 1.9 months, hazard ratio of 0.63. Again, when we looked at the squamous, it was essentially the exact opposite. The hazard ratio greater than 1, objective response rate, duration response, median PFS were all short for Dato-DXd, really emphasizing the patients with squamous histology are, when selected in an unselected manner, really not benefiting from Dato-DXd. We then looked at non-squamous histology that did not have actionable genomic alterations, because the question really was that benefit seen in the non-squamous population entirely driven by the actionable genomic alteration patients? And what we saw was no, that patients with non-squamous histology, even without actionable genomic alterations, had a PFS benefit for Dato-DXd, over docetaxel with a hazard ratio of 0.71. And so clearly the PFS benefit in non-squamous histology was for both patients with and without actionable genomic alterations. We then turned our attention to the second dual primary endpoint, overall survival. I presented an interim overall survival analysis in the intention to treat population, but there is a final overall survival analysis that hasn't occurred and will be presented at a future meeting. In terms of the interim analysis, we did see a trend in favor of Dato-DXd over docetaxel. This did not cross the pre-specified boundary for statistical significance. But when we broke it down by histology, again, what we saw was that the non-squamous patients are the patients deriving the greatest OS benefit from Dato-DXd over docetaxel.
So no, that was great, Aaron. Thanks. So Ben, I think I was surprised by this, the squeams. If we just did a CME not long ago, and we were talking that TROP2 is more highly expressed on adenocarcinomas of 100%. I think you actually said it was 92% for squamous cells. So not a lot of difference, right? So I'm a simple guy, and I would expect this to work equally in both. Yet in this study, squamous did actually, if anything, did worse with this compared with docetaxel. And we all agree we're not a big fan of docetaxel. Can you explain that, Ben? Do you have any thoughts on it?
Yeah, I think you're trying to seek revenge on me for the hard question I asked you when I was moderating. We don't know. We don't know the biological rationale why we saw this signal. And like many times in lung cancer now, what we do is we see the clinical observation first, and then we're going to have to work backwards to explain it preclinically. And that used to be the other way around, because the data is coming so quick and fast. So I don't think we have a good biological rationale as to why we saw this, but we need to do work. Question is, will we see the same signal in EVOCO-1, which is the same study, but using sacituzumab govitecan versus docetaxel? Fortunately, that trial also allows adenos and squames. So we'll see. It'll be really interesting if we see a different result from that. It may be more confirmatory if we see, hey, even with another TROP2 ADC, we don't see activity with squames. So I don't think we know, but it's early, and hope we get answers in the next 6 to 8 months here.
Yeah, no, and my only thought there is, both Aaron and I think, participated in the phase 1 study. We didn't talk much about it today, Pan-Tumor-01. And I admit I should go back and look at our own manuscript and see what percentage were squames. I'll be curious to see, was there just not enough squames? Was this a signal missed? Or is there something else going on that we don't understand? But Aaron, also, you mentioned before, talking a little bit about datopotamab plus durvalumab. And I know, Ben, you published a lot on that. As well, combining this with immunotherapy. What do you think the implications of that are, and how do you see this moving into the front line as we possibly combine checkpoint inhibitors with either carboplatin or immunotherapies like Durva?
Yeah, this is the kind of the key question here, is that we've seen single agent activity in the second line. How do we leverage that and combine it with either IO in the first line, or dare I say IO plus platinum in the first line, and replace our beloved pemetrexed for adenocarcinoma? So, I think there are two stories emerging here in the first line. We've seen encouraging activity, as Aaron mentioned, with datopotamab deruxtecan in combination with immunotherapy. We've also seen encouraging activity with sacituzumab govitecan in combination with immunotherapy, both in the frontline setting. The question will be, how does this move forward? And I think there are key studies that are answering this question. I think for the PD-L1 greater than 50%, the right trial is Pembro versus Pembro plus ADC, TROP2 ADC. And that trial is being done, and we'll see what that shows. Less than 50%, things get tricky. And how to design these trials is really important. The big question is, can we abandon the Keynote 189 or the 407 regimen for adeno and squames, respectively? Will the ADC do that? Will it just be ADC Pembro that's able to be, or at least equivalent to a 189 regimen? So you don't have to use the platinum or the pemetrexed. That can be replaced with the ADC. I think this is where things get extraordinarily tricky. Time will tell, but as Aaron briefly mentioned, we have seen encouraging activity with ADC plus immunotherapy. There's a biological rationale for this. Maybe there's synergy because ADCs work via ADCC, or antibody-dependent cellular cytotoxicity. So, look, we're at the alpha stage here. We're at the very beginnings of this story, and we'll have to learn more over the next 12 months. Guaranteed, if we had another of these in 12 months, we'd have an entirely different conversation.
And I'm sure we will do another one, hopefully together in 12 months, Ben. And I think for me, I'm looking at this a lot, and I do have some concerns, like moving to the front line. You start to not just talk about response rates, but you're also talking about tolerability. And I think we're going to be doing another one of these very shortly. That's not only just talking about efficacy, but really how do you manage toxicity. And it is my beloved pemetrexed. I've been using it for a long time. I actually did the phase 3 randomized study way before either one of you guys were around.
Dating yourself, Alex. You're
dating myself. I always do. I think it's a great drug. So how much better does it have to be? And what's the trade-off in terms of toxicity, right? And I think that's going to be a very important question. We only have about a minute or so left. So I think it's very exciting. And I think, you said it very eloquently before in a previous conversation, Ben, synthetic biochemistry for these ADCs is amazing. There are 3 parts. You can take any antibody, any linker, any warhead, combine them in all different ways, and you put it together. And there's a lot of these new targets. We have patritumab, we have TLOV, we have Nectin-4, we have tissue factor, we have a lot of these coming down the pike. Is there one, Aaron, you're most excited about coming down sooner? What are your thoughts on that?
Yeah, no, that's a good question. I was excited about the Herthena Lung 1 data that was presented at World Lung, the patritumab deruxtecan, specifically in the EGFR mutant population. I think the data there looked actually very similar to what we saw with Dato-DXd in the EGFR population. So there's some opportunities there. Teliso-V, I think, is an exciting molecule in the CMET space, using either the MMAE payload or the new molecule using a topo-1 payload. But I think that there's a lot of excitement across the spectrum of these ADCs. And I think that as we've all discussed throughout these the talks today, we're very early in development and really identifying which of these agents are effective, which patients are most likely to benefit. And then ultimately, if we are fortunate enough to get multiple agents in the clinic in the coming years, how do we sequence these things? Can we sequence these things? Which drug should we be using first? I think they're all exciting questions. I think they'll all be very difficult questions to answer, but there are more opportunities for hopefully our patients to live longer and better lives.
Yeah, it's truly a time, right? The number of drugs in development keeps exploding. The EGFR world with patritumab is going to get complicated. We have naked antibodies, we have bispecifics, so it's going to be really complicated. And you can just see it's going to be, how do we make decisions is important. So thank you for joining us for this segment on the impact of ADCs. Please be sure to click on the landing page for this activity to claim your continuing education credit, access supplemental slides, as well as other topic segments.
We hope you found this podcast useful and educational. To receive continuing education credit and to download your printable certificate, please go to the activity page at practice.cme.com to complete the post-test and evaluation to receive continuing education credit.